Hi everybody, and welcome back to our Chapter 3 discussion on neural control of exercising muscle. So to begin our discussion, we need to review the major divisions of the nervous system. Our two main divisions are the central nervous system, which is made up of the brain and the spinal cord, and our peripheral nervous system, which is pretty much every other nerve outside of these two areas. Our peripheral nervous system is then further divided into our sensory or afferent nervous system, and our effector or efferent nervous system. Again, our efferent nervous system is then further divided into the autonomic and somatic sections, and then the autonomic nervous system is divided into the sympathetic and parasympathetic divisions. This will become more important later in our discussion. So to start out, we need to discuss our structure and function of our nervous system. Our nerves, or our neurons, are the basic structural unit of our nervous system. They're going to have the same basic structure everywhere in the body. We divide the nerve into three major regions: the cell body or the soma, the dendrites, and the axons. Our cell body is going to contain the nucleus and is going to have cellular processes that radiate outwards. Our dendrites are going to handle the receiver part of the cell processing. These will carry impulses towards the cell body. Our axon will then act as the sender part of the nerve cell. This is going to start at the axon hillock and will end in branches, terminals, and neurotransmitters. These electrical signals will enable communication between the peripheral nervous system and the brain. However, for this to work, it must be generated by a stimulus. It also only works in one direction and is propagated down the axon. It must also be transmitted to the next cell in line. In order for propagation of a nerve signal to work, every cell must have a resting membrane potential. This is going to be the difference between the electrical charges outside of the cell and inside of the cell. A resting membrane potential is normally around -70 millivolts. This is because of the uneven separation of charged ions by the cellular membrane. This creates a polarized membrane. Also, why is it only -70 millivolts? This is because we have a high sodium ion concentration outside of the cell and a moderate potassium concentration inside of the cell. That means the inside of the cell is going to be more negative relative to the outside. This works because sodium channels are closed. This means sodium that wants to enter the cell cannot. This creates an electrical and a concentration gradient. Also, the potassium channels remain open. This means that potassium can leave and enter the cell via the concentration gradient. We offset this by potassium-sodium pumps. Whenever we change from the resting membrane potential, we typically are going through either depolarization or hyperpolarization. Depolarization occurs when the inside of the cell becomes less negative or more positive, meaning we go from -70 millivolts towards 0 millivolts. This is because sodium ion channels will open and sodium can rush into the cell. This is required for a nerve impulse to arise and travel down the axon. Hyperpolarization, on the other hand, occurs when the inside of the cell becomes more negative, going from -70 millivolts to -90 millivolts. This happens because more potassium channels open, and potassium can then leave the cell. This makes it much more difficult for a nerve impulse to arise and travel down the axon. The depolarization and the hyperpolarization within our nerve cells are going to create graded or action potentials. Graded potentials are going to help the body decide whether to pass a signal down the axon or not. This means that it can excite or inhibit a neuron. Action potentials, on the other hand, can only pass the signal down the axon and are only excitatory. Graded potentials only create localized changes in the membrane potential. This is going to be generated by the incoming signals from the dendrites. If it receives an inhibitory signal, there will be an efflux of potassium ions, which creates a hyperpolarization. If the signal is excitatory, there will be a sodium influx, which causes depolarization. If the graded potential is strong enough, it can lead to an action potential. How strong does this graded potential need to be, though? It needs to be strong enough to reach the threshold millivolts. The action potential will then be propagated down the axon, and the action potential will then be transmitted to the next cell. Our action potentials are going to involve rapid, substantial depolarization. It only lasts about 1 millisecond, and it always begins as a graded potential. If the graded potential at a cell reaches the threshold level, which is -55 millivolts, an action potential will occur. If the threshold millivolts of -55 millivolts is not reached, no action potential will occur. This is referred to as the all or none principle. Action potentials either occur or do not occur. What typically happens in the cell is that the cell will go from -70 millivolts upwards to 30 millivolts, and then back down to -70 millivolts again. The process of going from -70 to -55 millivolts, or reaching threshold, is going to be a depolarizing graded potential. This is caused by sodium influx. If we go from -55 millivolts to 30 millivolts, this will be caused by a depolarizing action potential caused by a sodium influx. Anything from 30 millivolts to -70 millivolts going back down to become more negative is going to be a repolarizing action potential, and this will be caused by a potassium efflux. This is an example of what an action potential will look like. If we start at the beginning, the resting membrane potential will be maintained by sodium-potassium pumps. Going through to depolarization, sodium ion channels will open, and sodium ions will move into the cell, therefore depolarizing it. These sodium ion channels will overshoot, and this will cause them to close, and potassium ion channels will begin to open to repolarize the cell. As the cell repolarizes, potassium ions will move out of the cell. This will then cause a hyperpolarization. This is due to the additional potassium ions moving out of the cell, hyperpolarizing it. The cell will then slowly return to its resting membrane potential. Early on in this period, the cell is less able to respond to another action potential. Action potentials, therefore, have refractory periods. There are two distinct refractory periods. The absolute refractory period is caused during depolarization. This is when the neuron is absolutely unable to respond to another stimulus, no matter how strong that stimulus is. That is because the sodium ion channels are already open and cannot open anymore. The second refractory period is the relative refractory period. This is occurring during repolarization. This is when a neuron response can respond, but only if it is a very strong stimulus. This is because potassium ion channels are open and the sodium ion channels are closed, but have the ability to reopen. The propagation of an action potential down the axon is sped up by myelin sheaths. Myelin sheaths are fatty sheaths that surround an axon that are produced by Schwann cells. These cells are not continuous and are separated by nodes of Ranvier. This produces salutatory conduction. This occurs by kind of having the signal hop from node to node down the length of the axon. A disease that is caused by the degradation of the myelin sheath is something called multiple sclerosis. The larger the axon diameter, the faster the propagation. So this means large myelinated axons are going to transmit signals faster than smaller or unmyelinated axons. Once the action potential reaches the end of the axon, it is going to reach a synapse. A synapse is the junction or gap between the neurons. This site will serve as the neuron-to-neuron communication. The action potential must therefore jump across the synapse. In order for this to work, the axon must send the action potential to the synapse, and then the synapse will send the information to the following dendrites. Your presynaptic cell must send things to the synaptic cleft, and then the synaptic cleft will send the signal to the postsynaptic cell. The signal will change form as it crosses the synapse. This form change will be in the form of neurotransmitters. This means our action potential will go from an electrical conduction to a chemical conduction, and then back to an electrical conduction. Action potentials can only move in one direction. That is because axon terminals create or contain neurotransmitters. These neurotransmitters are going to be our chemical messengers that hang out in the synapse. These will carry electrical action potentials across the synaptic cleft. These neurotransmitters will then bind to receptor sites on the postsynaptic surface. This will stimulate a graded potential in the postsynaptic neuron. The site of neuron-to-muscle communication is referred to as the neuromuscular junction. The specific neurotransmitter here is acetylcholine, and this is going to only be excitatory in nature. The postsynaptic cell in this case will be our muscle fiber. The acetylcholine will then bind to the receptor at the motor end plate on the muscle cell. This will cause a depolarization. This action potential will then move along the plasma lamina, down the T-tubules, and then undergo repolarization during the refractory period. There are over 50 known or suspected neurotransmitters in the human body. These neurotransmitters will fall into two major categories: small molecule rapid-acting neurotransmitters or large molecule neuropeptides that move slowly. Acetylcholine and norepinephrine are the two main neurotransmitters that govern exercise. Acetylcholine will stimulate skeletal muscle contraction and mediate parasympathetic nervous system effects, whereas norepinephrine will mediate sympathetic nervous system. Exercise is going to increase the rate of acetylcholine release. Peroxisome proliferator activated receptor coactivator 1-alpha, or PCG1-alpha, is going to increase the branching of the presynaptic terminals in our motor neurons. This will also increase the number of presynaptic vesicles that contain acetylcholine. Additionally, this will increase the number of acetylcholine receptors on the cell membrane and decrease the size of the motor end plate. This means that the more that we exercise, the more sensitive our muscle cells will become to the neurotransmitter acetylcholine. Our postsynaptic response means that our neurotransmitters will trigger graded potentials on the new cell. These graded potentials will either excite or inhibit the postsynaptic cell. Our excitatory postsynaptic potential is a depolarizing and excitatory response that promotes the action potential in the postsynaptic cell. This creates summation. When we receive multiple excitatory postsynaptic potentials, we create more polarizing or more depolarizing our postsynaptic cell. If we reach a threshold stimulus, an action potential occurs. On the other hand, the inhibitory postsynaptic potential is a hyperpolarizing and inhibitory response, which prevents an action potential in that particular cell. The summation of an inhibitory postsynaptic potential means that we will have a more hyperpolarized cell, which makes it much more difficult to stimulate that particular postsynaptic cell. Looking more into the general makeup and anatomy of the nervous system. Our central nervous system consists, as we've stated before, of the brain and the spinal cord. The brain itself is broken into four major areas: the cerebrum, the diencephalon, the cerebellum, and the brainstem. Looking at the brain, we can split the brain into two distinct hemispheres, the left and the right hemisphere. These two hemispheres are connected by the corpus callosum, which allows inter-hemisphere communication. The outermost layer of the cerebrum is known as the cerebral cortex. It consists of gray matter, which is non-myelinated nervous cells, and is the location of our conscious brain, which allows us to have our mind, intellect, and general awareness. This is further divided into four superficial or outer lobes: the frontal lobe, which houses general intellect and motor control; the temporal lobe, which has auditory input and interpretation; the parietal lobe, which has our general sensory input and interpretation; and the occipital lobe, which has our visual input and interpretation. There is also one deep central lobe called the insular lobe, which works with our emotion and our self-perception. The cerebrum holds a few regions of interest for exercise physiology. The first is the primary motor cortex, or the frontal lobe. This is where we have conscious control of our skeletal muscle movement. This is controlled by our pyramidal cells that will send information to the cortical spinal tract and then ultimately to the spinal cord, which will communicate out with the rest of the body. Our cerebrum also holds our basal ganglia, which is our cerebral white matter. These clusters of cell bodies lie deep in the cerebral cortex. They are going to initiate sustained or repetitive movements like walking, running, posture control, and muscle tone. Our cerebrum also contains our primary sensory cortex as part of our parietal lobe. The second part of the brain is the diencephalon. This contains two important regions called the thalamus and the hypothalamus. Our thalamus serves as our major sensory relay center, and this determines what we are consciously aware of. Our hypothalamus is going to be important because it helps maintain homeostasis and regulates our internal environment. Our hypothalamus works as part of our neuroendocrine control, meaning it works as part of our endocrine system, which we'll discuss in Chapter 4, and our nervous system. Our hypothalamus helps control appetite, food intake, thirst, fluid balance, and our sleep. It also regulates further control of our blood pressure, heart rate, breathing, and temperature. The third region of our brain is our cerebellum. Our cerebellum controls rapid and complex movements. It also helps to coordinate the timing and sequence of our movements. It also compares movements with intention and initiates connections and corrections. Our cerebellum also helps account for our body position in space and the status of our muscle tension. It is going to receive input from our primary motor cortex and help execute and refine our movements. Our next region of the brain is the brainstem. Our brainstem works to relay information between the brain and the spinal cord. It is made up of the midbrain, the pons, and the medulla oblongata. Our brainstem contains the reticular formation, which helps us coordinate skeletal muscle function and tone. It also works to control our cardiovascular and respiratory functioning. Our brainstem also has the analgesia system. This is where we interpret opioid substances that modulate pain. The brainstem releases beta endorphins here with exercise. This is what allows us to receive things like a runner's high after high-intensity physical activity. Our next region is the spinal cord. The spinal cord is going to be continuous with the medulla oblongata. It has tracts of nerve fibers that permit two-way conduction of nerve impulses. There will be ascending afferent sensory fibers, which will bring sensory information from the body up to the brain, and then descending efferent motor fibers, which will send control signals from the brain down to the body and the musculature. Looking deeper, we have the peripheral nervous system. Our peripheral nervous system connects the brain to the spinal cord. There are 12 nerves called our cranial nerves. These are connected directly to the brain. There are then a further 31 pairs of spinal nerves which connect directly to the spinal cord. Both of these types are going to directly supply nerve information to the skeletal muscles. Our peripheral nervous system is then divided into two major divisions, the sensory or afferent division and the motor or efferent division. Our sensory division works to transmit information from the periphery of the body back to the brain. There are going to be major families of sensory receptors. Our mechanoreceptors, which give us information about mechanical forces that impact the body. Our thermoreceptors, which regulate and give us information of temperature. Neuroreceptors, which give our brain information about pain. Photoreceptors that give our brain information about the light. And chemoreceptors, which give us chemical stimuli information in the body. There are three special families in the sensory division that work as sensory receptors that are particularly relevant in exercise physiology. The first are our joint kinesthetic receptors. These receptors are going to be sensitive to joint angles and rate of angle change. This will give us an idea of our joint position and movement in space. Our second special family are muscle spindles. These are going to be sensitive to muscle length and rate of change. This is going to give us information about how much muscles stretch. Our final special family are Golgi tendon organs. These are going to be sensitive to tension in the tendons of our body. This will give us a sense of strength of contraction. Looking into the motor division will work to transmit information from the brain to the peripheral nervous system. This is going to include two major subdivisions, the autonomic nervous system, which will regulate our visceral activity for our major visceral organs, and our somatic motor division, which is going to stimulate skeletal muscle activity. This will be the most relevant to exercise physiology. In the motor division, the autonomic nervous system will function to control the involuntary internal functions of the body. A few exercise-related autonomic regulations by the autonomic nervous system will be heart rate, blood pressure, and lung function. Our autonomic nervous system will be further divided into two other complementary divisions, the sympathetic nervous system and the parasympathetic nervous system. The sympathetic nervous system works as our fight or flight reflexes. This is going to prepare the body for exercise. Our sympathetic stimulation will increase our heart rate, blood flow to our working muscles, the diameter of our airways, referred to as bronchodilation, increase our metabolic rate, glucose levels, and free fatty acid levels, as well as our mental activity. Our parasympathetic nervous system, on the other hand, works as our rest and digest system. This tends to be the most active at rest and will oppose any sympathetic nervous system effects. When our parasympathetic nervous system is stimulated, this tends to increase digestion and urination, conserve energy, decrease heart rate, and decrease the diameter of blood vessels and airways. This is a good summary slide of the effects of the sympathetic and parasympathetic nervous system on the various organs of the body. I would take the opportunity to pause the video here and kind of read through this effect. The next component is sensory motor integration. This is going to be the communication and interaction between the sensory and the motor nervous systems. An example here is a stimulus is received to the skin by a sensory receptor, in this particular case a neuroreceptor or a pain receptor. The action potential will then travel through the sensory neurons to the central nervous system. The central nervous system will then interpret the information and then determine the motor response or the reflex to initiate the appropriate motor response. The motor action potential will then travel out of the central nervous system through the motor neurons. The action potential then reaches the muscle fibers and a response occurs. This is going to be typical of any sensory motor integration event. Additionally, sensory input can be integrated into many points in the central nervous system. The complexity of the integration will increase as we ascend through the central nervous system. The spinal cord, lower brainstem, cerebellum, thalamus, and cerebral cortex will all work to function as a sensory motor integration point. Our sensory input can also evoke motor responses regardless of the point of integration, whether it be the spinal cord, the lower region of the brain, the motor area, or the cerebral cortex. As the level of control moves from the spinal cord to the cerebral cortex, the movement complexity increases. Additionally, our sensory motor integration can do something called a reflex arc. Our motor reflexes are instant, pre-programmed responses to a stimulus. This response to the stimulus is going to be identical each time, and is before our conscious awareness of an event. An example of a motor reflex would be if you were to accidentally put your hand in a hot pan. You would not have to realize that you were in pain before your hand reached out and removed itself from the hot pan. This is because this impulse is integrated at a lower, simple level, typically in the spinal cord, before reaching conscious decisions in the brain. Additionally, within our sensory motor integration, we have two specialized types of cells, muscle spindles and Golgi tendon organs. Our muscle spindles are specialized intrafusal muscle cells. These are going to be different from our normal extrafusor muscle cells. These are going to be innervated by gamma motor neurons. These sensory receptors are going to give us information about muscle fiber stretch. If they are stretched, the muscle spindle sensory neuron is going to synapse with the spinal cord at an alpha motor neuron. This will trigger a reflex arc from muscle contraction. This is going to prevent any damage or further stretch within that particular muscle. This is referred to as a stretch reflex. Golgi tendon organs, on the other hand, are going to be sensory receptors embedded within a tendon. This typically is going to be associated with somewhere between 5 and 25 muscle fibers. This is going to be sensitive to tension that is built up in the tendon. It kind of works like a strain gauge. When it is stimulated by excessive tension, the Golgi tendon organ is going to inhibit the agonist excitation of the antagonist. This is going to present any excessive tension in the muscle, tendon, and other areas of that particular muscle cell. This is going to reduce any potential for injury. Finally, the motor response is going to occur because the alpha motor neuron will carry an action potential to the muscle. This action potential is spread to the muscle fibers of the motor unit. Fine motor control means that there will be fewer fibers per motor unit. Gross motor control means that there will be more fibers per motor unit. Fiber type will be homogeneous throughout a motor unit. This means fiber types are not mixed within a given motor unit. This means that type 1 muscle fibers will have a particular motor unit, and type 2 muscle fibers will have their own motor unit. Unit type either has type 1 fibers or type 2 fibers. This also means that motor neurons may determine fiber type. So, in conclusion, thank you so much for listening to Chapter 3, and I look forward to listening back to you guys in Chapter 4.